Good day, everyone. My name is Dr. Duyilemi Ajonijebu. I'm a human physiology lecturer and neuroscience researcher at Nelson Mandela University. I'll be speaking on early postnatal fostering, altered cocaine-induced inheritable epigenetic marks and non-specific chronic inflammation in drug-naive offspring mice. So, it's very important to recall that there's a previous study by Richards who reported that phenotypic variation is traditionally passed into components that are directed by genetic and environmental variation. And the line between these components is blurred by inherited epigenetic variation, which is potentially sensitive to environmental input. The figure to the right also shows the interaction between environment and addiction phenotype and how they are influenced by both genetic and non-genetic factors. On this knowledge, we previously demonstrated that cocaine consumption influenced characteristic changes in DNA methylation of CREM, that cyclic response element modulator and phosphate genes in the brain. Despite this evidence, there's the exact molecular and circuit-linked regulatory mechanisms that underline or underpin the transfer of cocaine-induced epigenetic marks to succeeding generation are not fully understood. This led us to hypothesize that parental cocaine exposure could impact on the immune response of drug-naive offspring, accompanied by alteration in the epigenetic machinery. We also hypothesized that cocaine-induced functional and neurochemical changes may be modified further by early postnatal fostering. And our main aim was to investigate whether parental cocaine exposure could impact on postnatal immunity and affect DNA methylation in specific promoters of cyclic response element modulator and phosphate genes in the prefrontal cortex of non-drug exposed offspring mice. We further sought to determine or investigate the overall impact of postnatal fostering on blood profiles, DNA methylation changes, memory competence, and locomotor behavior of the offspring mice. We obtained cocaine powder from the Forensic Science Laboratory through South Africa Police Services. The compound was further subjected to purity analysis using nuclear magnetic resonance. And the NMR spectra was recorded on Bruker-Evans instruments, and we used deuterated methanol as a solvent. And importantly, we were able to, we also used 9 to 10 weeks old C57 black cyst mice, which were obtained from Biomedical Resource Unit of the University of KwaZulu-Natal in South Africa. Both male and females were used. The females, which we call dams, they were given free drinking access to cocaine in the IntelliCage system. The IntelliCage system is an automated classroom apparatus which allowed behavioral phenotyping in rodents. And mainly the cocaine is 300 milligrams per liter dissolved in water. And they were exposed for 30 days in the IntelliCage. The males received alternate intraperitoneal injections of 10 milligrams per kg cocaine, or alternate, some of them also received, the control group received 0.9% saline for 11 days. We sourced for ethical approval through the Animal Research Ethics Committee at the University of KwaZulu-Natal. The structure of the IntelliCage is on the right, and with the various interior components. Now, the main experimental protocol. Basically, we mated male and female mice, adult mice that were previously exposed to drugs, and those that were not exposed to drugs were mated, so they were paired for mating. And as you can see in the structure, it's important to note that what we did was to the pairing mechanism involves is a 2-to-1 ratio pairing mechanism, such that we had 2 females paired against 1 male for mating. And if you recall, if you can see rather, as we have it on figure B, you see that we have a group of animals, both male and females, that were not exposed to drugs paired together for mating. We also have another group where we have only the males that were exposed to drug, and that is the ME group. And then we also had another group where the male was not exposed to drugs, but the females were the only ones exposed to drugs and they were paired for mating. That's the FE group, that's female exposed. Then we also had male and female exposed, where both male and females were exposed to drugs and paired also for mating. Then after mating, at parturition, the offsprings were, half of the offsprings were also given over to fostered mother to nurture them to maturity. And the fostering process is that we collected half offspring from FE group and gave it over to a mother in the NEP group, and also vice versa. The same thing applies to MFE and ME group. Then also we collected blood from these animals, both the parents and especially the offsprings rather, and we analyzed the full blood analysis was done using Belka Coulter counter, or it's a blood analyzer. And we specifically looked at the total number of counts for neutrophil and lymphocytes, and subsequently we looked out for the ratio, which is called neutrophil lymphocyte ratio. Then behavioral assessment was also done at postnatal day 44 to 47 days. The animals were sacrificed at postnatal day 48, and the brain region that was removed is the prefrontal cortex, and we subjected that further to DNA methylation. And for concerning the results, we observed that at postnatal day 44 to 47 days, when we performed memory, functioning, and locomotor activity, we observed that neither the parental cocaine exposure nor fostering affected the locomotor behavior of the offspring mice. Concerning the memory performance, we also basically, when we assess the memory competence using the NOR, that's Novel Object Recognition Test paradigm, we noted that both NEP and FE non-fostered offspring mice, non-fostered offspring mice and their counterparts and their fostered counterparts, as you can see, the NEP and also the FE, both the non-fostered and their fostered counterparts, they attained 50% discrimination. So memory performance was good in these two groups of offspring. Now, in the second groups of offspring, we discovered that there was particularly the cross-fostered ME offspring was memory challenged, as they failed the performance test because they could not achieve a 50% discrimination index. And further to that is that we also noted that both MFE non-fostered as well as MFE cross-fostered, these two groups of animals, they were severely memory challenged. So memory performance was apparently poor in both groups of animals because none of these groups was able to achieve 50% discrimination index. So we concluded that drug-naive offspring mice raised by either biological or non-biological mothers exposed to cocaine are memory challenged. Then we also look at metallation changes in the prefrontal cortices of both the parent mice as well as the offspring mice. And from what we were able to gather so far, we see that there was significant hypomethylation in the promoter regions of CREM as well as FOSB in both the cocaine exposed dams as well as that of sires, as you can see in this structure. Now, the same pattern of hypomethylation was also seen in the FE, ME, and MFE groups of offspring in their brains as well. And same also for FOSB. So looking at the cross-fostered group, cross-fostering or early postnatal fostering apparently had no impact on these DNA methylation changes. And then we concluded that patterns of DNA methylation expressed in the prefrontal cortices of drug-naive offspring mice are similar to their progenitors. Then looking at the blood profile, it's very important to note that that there's a study by Soda et al. In 2020, published in 2020, that supported the use of NLR, which is called the neutrophil lymphocyte ratio, to be used as a marker of non-specific inflammation. So on that note, we examined the transcriptional impact of parental cocaine exposure on circulating inflammatory cells, neutrophils, and lymphocytes in this case, in the drug unexposed offspring. And what we eventually settled on is that when we look at the neutrophil lymphocyte ratio, there was an elevation in the non-cross-fostered ME and FE groups, as you can see in these two groups, as compared to the NEP non-cross-fostered group. And we also saw that there was the lack of change in the MFE group could possibly be explained, by improved adaptive responses or coping mechanisms in the group of offspring as compared to genetic processes and favorable maternal-fetal immune interactions. However, activation of immune response in the offspring to parental cocaine exposure was also not affected by early postnatal fostering, as you can see in this structure, in this figure. So we concluded that elevated neutrophil lymphocyte ratio in non-cross-fostered ME and FE offspring mice confirmed non-specific inflammatory changes in the blood of drug-naive first-filial generation progenies in response to parent mice cocaine exposure. And we concluded that our data showed that indirect exposure to cocaine could lead to non-specific inflammation and some epigenetic marks associated with cocaine exposure, and these were passed down to drug-naive descendants. We also conclude that transcriptional changes mirrored by alterations in the epigenetic machinery, domicile in prefrontal cortex appears also reversed by early postnatal fostering. Finally, there's an association between cocaine in mediated epigenetic molecular events and impaired cognitive performance of drug-naive descendants. Future directions or future studies rather can look at or confirm CREM-mediated, CREM-mediated parent-offspring transmissions by looking at the epigenetic profiles in the sex cells of cocaine-exposed mice. These are my references, my acknowledgements. I want to say thank you very much for your time.